But by far, the most important part of an enzyme is what's called the active site. And the active site is what actually binds to the substrate or the reactant. Now, generally speaking, the active site on an enzyme, so this is a three-dimensional model of some ambiguous enzyme. The active site that's binding to whatever substrate we're trying to manipulate is generally small relative to the rest of the enzyme. And the rest of this enzyme represents a scaffolding or a structure to produce the right kind of active site. There is on the substrate, whatever it is you're trying to manipulate, what's called a binding site. And that's the area that the enzyme is going to attach to. And it's like a lock and key mechanism. The active site has to have a shape that accommodates the binding site of the reactant. Okay, are we okay with this so far? So this shows you some enzymatic reaction. What needs to happen is we need to take these two substrates, those two reactants, and we need to combine them into a single thing. We could say this is some photosynthetic process where we're taking a carbon dioxide, and we're attaching to it some other hydrogens and oxygens and carbons and things like that. So we're going to take these two substrates and we're going to stick them together, and a single enzyme is going to catalyze that reaction. And it has an active site that accommodates the carbon compound and perhaps the oxygen and hydrogen compound. So that enzyme connects to both. It does something biologically magical to them. It causes them to be stuck together so that the product is the combination of these two things, endergonic or exergonic reaction. What do you think? Yeah, it's endergonic because we're taking two simple molecules and sticking them together into something more complex. So that's an endergonic reaction. Now, had it happen in the opposite direction, which is metabolism, right, where we're taking this one substrate and we're breaking it into two, that would have been exergonic. Generally speaking, chemical reactions occur in series called biochemical pathways. So when we talk about photosynthesis and cellular respiration, that is inherently a biochemical pathway, where there are going to be a whole bunch of enzymes in sequence that take substrates and manipulate them a little bit. And you can think about biochemical pathways as like an assembly line, like Henry Ford's great invention, right? Where you start off with a reactant, and some individual does something to it. And then they take their product and they pass it to the next individual that does something to that, and then passes it to the next individual. So it's a very sequential and intentional sort of pathway. In cellular respiration, in metabolism, what we do is we take a sugar molecule, and we take that sugar molecule and we break it into 2 3-carbon chains called pyruvate. That's the first series of reactions. Then that product moves to the next step, and we take those 2 3-carbon chains and we completely break them down into individual carbon dioxide molecules. And we generate a whole bunch of protons. And those protons are passed off to the next series of reactions. That from those protons and the energy that those protons are generating, we make a whole bunch of ATP. So, actually, I'm going to blank this out really quickly. We've talked about changing the shape of proteins before. Now, changing the shape of a protein is called denaturing it. And how is it in the culinary arts that we denature proteins? We cook it. That's a primary way, right? We apply heat. We know that heat can take any number of molecules and change their shape. Okay, so temperature is a huge way to change the shape of a protein. How else can we do it?
We talked about like pressure and like tenderizing.
Yeah, exactly. So we can physically strain a protein and we can cause it to lose its shape. Yeah. How else can we do it? What's that? pH, exactly right. We can acidically change the shape of a protein. That's what we do with marinades, right? We take a marinade and we apply an acid to it, and it cooks the meat, essentially. It causes those proteins to lose shape. Yeah, go ahead.
What was the second thing you said that was, so the heat and temperature can change it, and then the acid
Pressure
pressure.
can change it, yep, and acidity can change it. And what's cool is enzymes do basically all of those things. An enzyme can take a biological molecule and it can increase or decrease the acidity, the pH, and cause a chemical reaction to occur by changing the pH within the active site. Or it can physically strain a molecule and catalyze a reaction that way. But those proteins, because they are proteins, are inherently susceptible to those physical characteristics as well. What's interesting, though, is pH and temperature have a different effect on enzymes. So if we were to, you'll have to think back to our enzyme lab where we were looking at liver. If we were to graph the effects of temperature, it would look something like this. Where on the x-axis down here, we have temperature. And we're going to go from 0, let's say, to 100 degrees Celsius. And then along the y-axis, we have enzyme activity. Now, we compared a couple of different temperatures. So we took our enzymes and we froze them a little bit in an ice bath. We heated them up to 37 degrees, and then we cooked them, right, in boiling water, which is 100 degrees Celsius. And that wasn't perfect because we weren't able to measure what happened at the temperatures in between those three discrete points. But if we were, then enzyme activity would look something like this. How would you interpret those results? What does that mean?
That you activated the protein, the enzymes.
Okay,
For whatever purpose you're recommending.
Okay, so by activation, I assume you mean that enzyme activity is increasing. Okay, so with increasing temperature, we have increasing enzyme activity until that point, at which point what happens?
And denature.
Okay, so the protein gets denatured, the enzyme gets denatured, and enzyme activity drops. At this point, that protein has lost its shape, right? But why the increase in enzyme activity with temperature, do you think? Yeah, Carly.
Because it's going through that process, that biochemical pathway, so it's using energy to get to that point, and then once it's changed, does it have anything?
I like the way you're thinking about it with using energy. And so what about this suggests, or where is that supply of energy increasing? What's the, oh yeah, go
I was
ahead.
gonna say, isn't it increasing to continue to try to maintain its shape, which is why it takes a lot of energy to
Right.
denatured.
Okay, I like it. So enzymes don't necessarily have to try and maintain their shape. It just happens because of the chemical bonds within those. But nothing's affecting its shape up to this point, right? It's maintaining its shape. It's able to catalyze that reaction. It's just the efficiency with which it's doing it is increasing with the increase in energy or temperature. So it's odd for us to think about enzymes as, I guess, biological entities. Let's remember, though, that enzymes don't actively seek out and find some substrate on which to act. Substrates come across enzymes and enzymes across substrates randomly. It's just a product of some random event where these two things happen to be at the same place at the same time. And that's a product of how much they're moving. And so as we increase temperature, what we're doing is we're increasing the movement of both the substrate and the enzyme, and it's more likely for them to come together. So we're increasing the amount of energy, right? But really what we're doing is we're increasing the rate of movement and interception between both enzyme and substrate. pH, however, has a very different... Oh, and I should mention, and my x-axis is going to be all off, but for us, this optimal enzyme temperature right here is about 37 degrees Celsius. And 37, does anybody know why that's significant?
Some temperature of your body.
Yeah, body temperature, right there corresponds to 98.6 or whatever body temperature is in Fahrenheit. Okay. So that's for most of our enzymes. But there are some organisms that have enzymes that are optimally active way down here to the right. I'm going to extend out this x-axis a little bit, so it's a little bit more symmetrical. There are some organisms that have an optimal temperature that looks like that approaches 100 degrees Celsius. And these are a group of organisms that we talked a little bit about in chapter 1. They are the Archaea, or extremophilic bacteria type organisms. So there are organisms that live in thermal hot springs, right? Like in Yellowstone. And those organisms, because they live in such a hot environment, have to have enzymes that don't denature at 37 degrees Celsius. They need to be optimally active at much higher temperatures. Okay. So if we were to compare that to the effect of pH on enzyme activity, the graph would look quite different. So here we're going to have pH, and we'll have 7, 0, 14, something like that. And then this is enzyme activity. So you guys, if I ask you to reproduce these graphs on an exam, I'll want the axes labeled, okay? So generally speaking, there is some optimal enzyme activity at the pH of a little bit over 7, because that's our blood pH. It's like 7.3 or 7.4, something like that. And then around that optimal pH, there is a bell-shaped curve. So somebody interpret those data for me.
Does it just mean that the chemical reaction lasts longer?
So, it doesn't necessarily mean that it lasts longer, but there is a symmetrical effect of divergence from pH, optimal pH. So what's happening here?
We know that the activity is decreasing as the pH increases past 7. But
What happens to it as it decreases past 7? what does that graph suggest? Because it's the same, right? So what you're saying is, as we go past 7, as we're diverging from this optimal point, enzyme activity decreases, right? That's just totally true. What happens as we become more acidic? It decreases as well. So as opposed to temperature, pH has a symmetrical effect. It doesn't matter if it's becoming more acidic or more basic or more alkaline. That divergence from the optimal pH is going to have the same effect. Or said differently, if enzymes are exposed to an increasingly alkaline solution, they denature. And if they're exposed to an increasing acidic environment, they also denature. There's some enzymes, however, that have an optimal pH that's somewhat close to 2. So their enzyme activity looks something like this. Could you think of enzymes that have an optimal pH that is very acidic? Where would those be active, do you think?
Would it be like plants that have more acidic, like oranges and
Okay, sure, that live naturally in acidic environments. I love it. Citric acid is a really, really acidic compound.
Can
How
they
about-
also be in us as humans, like
For
our
sure.
stomach?
Absolutely. Yeah, so think about this, you guys. We have this lumen inside our body, right, that runs all the way throughout our body. And it is incredibly low pH. And it's low pH because we know that low pH denatures proteins, right? I mean, that's why we marinate stuff. But we also have proteins that we don't want to have digested and denatured. Those are the enzymes that are catalyzing lots of these reactions. So our stomach enzymes tolerate a very, very low pH. Excellent. So these are those same graphs. Again, on the top we have the effective temperature on enzyme activity. So we have, for us, right, this increase in enzymatic activity up to the optimal temperature, which is somewhere around 37 degrees. And then it drops way off. Because it's at this point that that enzyme becomes denatured and loses its biological activity. Thermophilic archaea live in much warmer environments, and they don't have that denaturation until a much higher temperature. And then acid